morning to you and my fellow classmates. My name is Leesha. My teammates are Nalvinna, Samita, Samundeswari and Sherlina. Today we're going to present about our mini project, which is the making of histopathology slide. Histology explains the tissue structure and inner processes that impacts the tissue differently. Light and electron microscopy techniques are usually used to see the tissue and usually taught separately. These are the few steps involved in preparing the slides for histology technique. The first step is specimen lodging. Sample lodging is the point where histology laboratory receives the samples, receiving specimens and order request test form, both which are immediately time-stamped to record the time of arrival, and the data entry of all test orders are associated with the specimen. These are the procedure involved of dissection of the mice that has been done in the lab. The second step is tissue grossing and fixation. The process of going through the sample and identifying and measuring the tissue is tissue grossing, whereas the fixation is also synonymous with the concept of preservation. It stops any metabolic activity that could take place in the tissue.
Thank you so much, Leeshaalenee. So I will be proceeding with tissue processing. So basically in tissue processing, there are four main important stages, which is dehydration, clearing, infiltration, and impregnation. So basically dehydration, the main purpose of it is to remove the water in the tissue sample, because in paraffin wax is hydrophobic. So the majority of tissue water must be eliminated before it's waxed. So it starts from 50% alcohol and ends with 100% of alcohol. The next stage in tissue processing, The next stage in tissue processing is clearing. So basically the main purpose of clearing is to remove the alcohol used in dehydration process. So this alcohol is replaced with xylene, because xylene is used as clearing agent in this process, and it also helps to make the tissue transparent so that it's easier for the paraffin wax to be infiltrated. The next stage in tissue processing is infiltration. Next. Infiltration is to introduce the tissue sample with paraffin wax so that later on it will be even more convenient to proceed with embedding. So impregnation is to ensure that the last stage in tissue processing is impregnation. So basically impregnation is to ensure that the tissue sample is totally 100% pure, infiltrated with the paraffin wax. So that is the four main stages in tissue processing. Next. So this is the procedure and steps that is done in the laboratory. It starts from dehydration, clearing, and then it ends with infiltration and also impregnation. Next.
Embedding process. Eyes, tissues or the specimen are enclosed in the mass of the embedding medium using a mold. Since the tissue blocks are very thin, they need a supporting medium in which the tissue blocks are embedded. The choice of embedding media depends on the type of microscope, type of microtome, and the type of tissue. For example, like hard tissue such as bone, or soft tissue like liver biopsy. Next slide. There are few embedding media as far as we use in the lab, which is a paraffin wax with a higher melting point, which is 56 to 62 degrees Celsius, is used for embedding. The molten wax is filtered inside the oven through a coarse filter paper into another container. This will protect the knife edge, and others are carbo wax, epoxy resin, gelatin, and also agar embedding. Next slide. As for the methods of embedding, the first step is first we open the tissue cassette in order to check whether the tissue pieces are still present. And then the second one will be we have to select the mold, and there should be sufficient room for the tissue with allowance for at least a 2 micrometer surrounding margin of wax. The third step will be we fill in the mold with paraffin wax, and then using warm forceps, we'll select the tissue, taking care so that it does not cool in the air at the same time. Then we move on to placing the tissue in the mold according to the site to be sectioned. We'll chill the mold on the cold plate. More paraffin will be added into the mold to fill the cassette and mold. And after that, we'll cool the block on the cold plate. Lastly, we will remove the block from the mold. Next slide. As for the types of mold, most of the laboratory use plastic embedding rings now because it's inexpensive, convenient, and also support the block during the sectioning and are designed to fit in the microtome. The disadvantage of this method is the space required for storing is small. The Mark II system has provided a cassette to hold tissue during processing and has a stainless steel lid on the plastic cassette. The advantage of this cassette is that thin, so less wax are required and the space required for filling the block is less. Next slide. Okay, moving on to the orientation of different tissue. During embedding, the orientation of tissue is very important. Correct orientation of tissue in a mold is the most important step in embedding. Incorrect placement of tissues may result in diagnostically important tissue elements being missed or damaged during the microtomy. That is why correct orientation of tissue in a mold is the most crucial step. That's all from me.
Next is step 5, which is sectioning. Sectioning is a process done after producing a block during the embedding process. This process is to produce a tissue ribbon that will be easy for the pathologist to look for the details of the sample. And it requires 3 parts to prepare a section, which is trimming, sectioning using a microtome, and prepare a section. The sectioning process is done by preparing the frozen tissue block, the tissue flotation water bath, and also a slide warmer. Trimming is a process where the excess of the paraffin wax from the tissue block is cut into a trapezium shape. Next. This is the step that require in sectioning, where the first, as we can see in the flow chart, the first one is to cut the tissue block using a microtome. Then it will form a tissue ribbon and we put it at the flotation bath, and then we fetch the tissue ribbon with slides, and then we put the tissue slide at the stretching table, and lastly we air dry the tissue slide. Next. The video shows how to cut the tissue block into a trapezium shape, and the rest of the sectioning process, how it is done. Next. Next step is staining and mounting, which is used to view the details of the components through the microscope. And the purpose of the staining is to highlight the structures of microbe for viewing, and hematoxylin and eosin are 2 dyes used for the staining. Hematoxylin is a purple color dye that used to stain the nucleus of the tissue, and eosin is a pink color dye which used to stain the cytoplasm of the tissue. And chemical used in the H&E staining is xylene, alcohol 100%, 95%, 80%, and also 70%. And after the staining, the slide will be mounted with DPX and covered with cover slip. Next. Figure 31 shows the slide after being stained with hematoxylin dye, and figure 32 shows the slide after being dyed with eosin, and figure 33 shows the slide after mounted with UPX and placed on a stretching table. Next, I will pass to Sherlina.
Hi, my name is Sherlina and I will be presenting on step 7 and step 8. So for step 7 is microscope slide moving. So the slide will be viewed under the microscope after demounting, and during viewing, few troubleshoot have been encountered, and the slide was divided into 2 categories, which is good and bad slide. So in slide viewing, we have used 2 different microscopes, which is light microscope as well as the comparison microscope. So for the step 8, which is labeling, recording data and reporting, the microscope slide will be labeled with the respective tissue, and the observation of the microscope viewing has been recorded and recorded. So for the next slide, this slide shows the good slide which we have used the hematoxylin and eosin staining. So for the nucleus color with blue color, and the cytoplasm was color with pink color. So as you can see in this slide, they were glomerulus has been circled, and the Bowman space also can be identified from the slide. So according to a good slide, there are 2 characteristics for a good slide, which is a good staining. It can observe the cells clearly. It does not have any damage of cell spot or even any microtomic artifacts. So these are the characteristics of the good slide. And for the poor slide, it also is a bad slide. So according to this slide, the bad slide is the kidney slide, which we observe under 5 times magnification under comparison microscope. So the microtomic artifacts that have been found in this slide was formation of bubble, section disintegration on water bath, and also the knife lights. So the first microtomic artifacts is formation of bubble, which caused by the trapped air during the mounting. So for the resolution, we have to press the cover slip to remove the bubble during mounting. And the second one is the section disintegration on water bath. This is caused by water temperature is too high that could melt the wax, and the resolution is to decrease the temperature, which we have to maintain with the ideal temperature. And the last one is the knife lines. This is caused by the foreign materials in tissue mixed in the blade or dull or dirty blade. So as for the resolution, we need to change the blade and do not introduce tools to the edge of the blade. So these are the differences between the good slide as well as the poor slide. So as for the conclusion, histology technique is a study of the microscopic anatomy of the cells as well as the tissue of plants and animals by using light microscope as well as electron comparison microscope. So all the cell must be grown, fixed, undergo tissue processing, embedding, sectioned, stained, and mounted before observed under the microscope. So histology is very useful in studying the physiological function of the cells in the body, whether in human, animals, or in plants. So in the form of histopathological, it is useful in the diagnosis of disease that involve changes in physiological function as well as the organ. And histology also is an important tool of biology. So that's all from us. These are the references we have used. So thank you.